Now we're about to start talking about gene therapy. Gene therapy being a practice that you can be able to use it and edit genes. Okay, so we'll talk about gene therapy. But before we talk about gene therapy, it's good for us to refresh our knowledge on what a gene is. So I want to now talk about the role of a gene in the synthesis of protein. Okay, we'll need all this knowledge later on. So genes actually play a very central role in production of proteins. So they act as a blueprint for building every protein in the body. Okay, so what's the step by step to how a gene is involved in protein formation? So transcription, so DNA to mRNA. So inside the nucleus, a gene's DNA sequence is transcribed into messenger RNA. Okay, now what? This process is carried out by the enzyme RNA polymerase, which reads the DNA, then creates a complementary RNA strand. The RNA processing in eukaryotes. So the initial mRNA, called the pre-mRNA, is now modified. So introns will be removed while exons are spliced. Then what next? Okay, so I think basically the steps complete. All right, I'm coming. All right, so I went and actually got the full lecture. So the mRNA will now be processed in eukaryotes, and it will undergo processes like splicing, capping, polyadenylation to become mature. They're ready for translation. So translation is when the mRNA is now converted to protein. So it travels to the ribosomes, which is the cell's protein factory. So the transfer RNA molecule brings amino acids to the ribosome, matching their anticodons to the codons on the mRNA. So the ribosomes link the amino acids together in the correct order to form a polypeptide chain, which folds into a functional protein. So protein folding and function is after translation, the polypeptide may undergo further folding and modification. So that's what actually becomes a functional protein. All right, so that's what we got.